Let us continue with our discussion of gene regulation and gene expression in Chapter 13. Specifically, we were focusing on how messenger RNA is translated into a protein. During transcription, the nucleotides in DNA are read by enzyme RNA polymerase to produce messenger RNA. Transcription has 3 steps: initiation, elongation, and termination. Let's watch a video of messenger RNA synthesis, transcription.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
As mentioned previously, transcription has 3 stages: initiation, elongation, and termination. 3 steps of transcription correspond to the 3 major parts of most genes in eukaryotes and prokaryotes. A promoter region at the beginning of the gene marks where transcription is to be initiated. The body of the gene corresponds with where elongation of the RNA strand occurs. And a termination signal at the end of the gene marks where RNA synthesis is to stop. Let's watch a video of transcription.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Transcription begins when RNA polymerase binds to the promoter of a gene. The enzyme RNA polymerase synthesizes RNA. RNA polymerase binds to the promoter region at the beginning of a gene. The promoter consists of a site that binds RNA polymerase and one or more regulatory sequences. When RNA polymerase binds to the promoter, the DNA is unwound and the strands are separated at the beginning of the gene sequence. In step 2 of this figure, elongation generates a growing strand of RNA and RNA polymerase synthesizes a sequence of RNA nucleotides along one of the DNA strands. The strand that it is actively reading is called the template strand. RNA polymerase travels along the DNA template strand, starting at the 3' end of a gene and moving to the 5' end. The bases in the newly synthesized RNA strand are complementary to the DNA template strands. Starting at the initiation end, the forming RNA strands drift away from the DNA template strand, while the RNA polymerase holds the forming end to the template. As the RNA strand leaves the DNA strand, the helix reforms. Termination, which is shown here in step 3-4 of this slide. Termination stops when RNA polymerase reaches the termination signal. RNA polymerase reaches a termination sequence, releases the completed RNA, and detaches from the DNA. RNA polymerase is then free to bind the promoter region of another gene and to synthesize another RNA molecule. This figure is showing a strand of DNA, which is in blue, that is being transcribed in the RNA by RNA polymerase. You can't see the RNA polymerase enzyme, but you can see the RNA being made in the reddish, pinkish purple squiggly lines coming off the DNA in the center. This slide is showing transcription and translation in prokaryotic cells. Because prokaryotic cells do not have a nucleus, their DNA is in the same region of the cell where the ribosomes are. So a lot of times with prokaryotic cells, such as bacteria, transcription and translation will be occurring at the same time. After messenger RNA is made, it has to be processed before it can leave the nucleus and make a protein. In eukaryotes, a precursor RNA is processed to form messenger RNA that is translated into protein. Each gene consists of 2 or more segments of DNA that encode for a protein called exons that are interrupted by other segments that are not translated called introns. Transcription of a gene produces a very long RNA strand that contains the introns, which is not going to be used for the protein, and the exons. This long strand, which extends beyond the first and last exons, is often called precursor messenger RNA or pre-messenger RNA. More nucleotides are added at the beginning and end of the pre-messenger RNA molecule to form a cap and a tail. The cap and the tail assists with moving the RNA through the nuclear membrane to the ribosome and prevent cellular enzymes from breaking down the RNA. Enzymes in the nucleus will cut out the introns and splice together the exons to make true or mature messenger RNA. The messenger RNA will then exit the nucleus through a membrane pore and associate with a ribosome. There have been many debates as to what the purpose of the intron is and why you have this intron-exon gene structure. Through alternative splicing of the exons in a gene, a cell can make multiple proteins from a single gene. Alternative splicing represents an exception to Beadle and Tatum's one gene, one protein relationship. A second function for the presence of the intron-exon gene structure is that fragmented genes may provide a quick and efficient way for eukaryotes to evolve new proteins with new functions. If breaks in chromosomes occur in introns, exons may remain intact and be spliced to other chromosomes in a way that produces a new useful protein. Now let's watch an animation on how RNA is processed.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called RNA splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or SNRPs. SNRPs are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of SNRP binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional SNRPs then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop. This also gathers all of the SNRPs together as the completed spliceosome. The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the SNRPs are released.
Question 11. Which of the following is not found as part of mature RNA molecule? Choose the correct answer. Now let's take a look at how is messenger RNA translated into a protein. During translation, messenger RNA, transfer RNA, and ribosomes cooperate to make proteins. Like transcription, translation also has 3 steps: initiation, elongation, and termination. Let's take a look at translation, also known as protein synthesis.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.